Now?
Now it's good.
Well, I think this is the last lecture of the day, a really long day. And this presentation is related with immunity, but focuses in aquatic organisms. But different to Sergio, I think it's better if you ask during presentation, because with that also I can calibrate the detail of the different slides. We will go really deep in the molecules or in the cells, so this will be more in the surface. So I need to know who is in the chat, because I will ask. I know that for many people that is a nightmare, but for me it's fine. So when you read the term immunity, what is the first idea that is in your mind? Edison, are you here or not? No, Edison is not here. Deborah? No, I think we only have ghost students.
I suppose Deborah should be there. She has a good English. Victor is
The coffee was very long.
maybe so.
Well, I know Victor is here.
Yep.
Yes.
Yeah, but immunity, I think for many people, they read immunity and they think in, I don't know, attack, aggressive response, find a pathogen or destroy it. But I think also one of the main relevant concepts of the immunity is the homeostasis, because at the end, and mainly in fish, homeostasis is the key. And for instance, I will put, well, that is right now, there is more information about the immune homeostasis, especially related with health and diseases, because the immune response can be is aggressive against a problem, but also is very aggressive against the host. And that balance can be the difference between the survival or the mortality of the fish and other aquatic organisms. So this part of the presentation is an invitation to think in the immune response, not only against something, also against the balance. And it's one of the main systems involving the homeostasis. But when we say that this is a system, why? Why we can say that this is a biological system? I put this definition because I like it a lot, because it say that run smoothly. When the response is balanced, we don't know that the immune system is working. But when we have difference, I don't know, dysbiosis or an aggressive immune response, and we are infected, we know that the immune system is there. Question until here? Otherwise, this will be a huge monologue for 1 hour. Well, as I say, this is a system, and you can see how is the evolution of this system. There is a problem that the immune response is always described from the mammalian point of view, or from higher vertebrates, but the immune system is very old. For instance, here you can see porifera, and they are very relevant because in this kind of animal, we saw the first immune cells. For instance, the archaeocyte. Archaeocyte mean all cells, and they are the first one with phagocytic capacity. And phagocytic capacity is a factor process for the innate immune response. So from here, we can start to see how the immune response start to develop until vertebrates, and in fish. Starting with fish, we saw the other part of the mechanism that is the adaptive immunity, until homoterms shows us the mammal, but also the birds. So here you can see a progression of the immune response. This is the same idea, but I think maybe it's more friendly to see. This one includes plants, because also there is several mechanisms describing plants. For instance, the microbial association for molecular pattern were first described in plants. And also there is several molecules that start to be described in plants, and then they were described in animals. Well, the immune system is a system because has different organs. Each organ has different tissues, and we can find specific cells. That is why it is a system. And here in the right, you can see different cells, epithelial cell, goblet cells. These are part of the innate immunity. This one is a barrier. The other is secreting components such as mucins. You have also lymphocyte, part of the adaptive immunity. Many of these cells are in the blood, which is a specialized connective tissue. Then you have another structure, and also you have the coordination of organs. And for instance, in fish, there are some difference because in fish we don't have the lymph nodes, but we have primary and secondary lymph organs. So all these organs and tissues are connected, and at the end, the immune response is a very integrated one. That is why we can describe organs. Good? No questions?
No, it's clear.
Yeah. First, the hematopoietic process is very similar with mammals. You can see here that we have, I will put the laser. Here we have several cells that also are in higher vertebrates, but the difference is where. In fish, the hematopoietic process is mainly related with head kidney. Otherwise, in some specific fish, also the thymus can be involved. But for many fishes, the hematopoietic process starts in the head kidney as a primary lymph organ. There, they can be mobilized to other organs, and they can be resident, for instance, in the gut, but also in the spleen as a secondary lymph organ, and the mucosal associated lymph tissue that we have in the gills, in the skin, and in the gut. Here you have an overall view of the cells and the tissues and the organs. So with that, I reinforce the idea that the immune response is a system. Here in the left, you can see the mucosa associated lymph tissue that right now they are really relevant to many immunonutritional activities and approaches. For instance, using functional feed, their main focus is the gut, but also this gut can be linked with other tissues and with other organs. For instance, the gut with the liver, the gut with the head kidney, the gut with the gills. And that is really important to describe all the different cells, because one thing is determine modulation of something, but who is doing the final job? And those are the specific cells and the specific effector molecules. This is a classical figure of immunity 101 that you can find that describe the overall view of the immune response. But always remember that this is from higher vertebrates, but that can be applied to fish. Here we can see the two main mechanisms, the innate and adaptive, both with cellular responses and humorals, the antibodies. When you hear humorals, they are molecules. So it's the fancy way to say, there is molecule involved, is humoral response. What is the difference between both? What is the most important factor to define which one is acting? And what do you think? Another ghost student. I have the complete list. Anthony, I already asked Deborah, Maximiliano, Samira. No, ghost student.
Samira has problem with her mic.
Hi, I'm listening very carefully.
Yeah.
But
so what is the main factor to define which is innate and what is adaptive in this kind of figure? How you determine which type of immunity is acting against in this case, against an infection?
Well, innate immunity is related with the microbes outside the cells, and adaptive immunity is occurring inside the cell. As long as
Not
I
really.
can see, as long as I can see, because my topic of research is very far from
Yeah, but if
this
you see the
topic.
figure and this one, the main variable is time. It's time of
Okay,
exposure,
yeah.
time of infection. The innate response is faster, just need a couple of hours. While the adaptive immunity, they will need more time. Why? It's because this type of mechanism needs to coordinate these ones. And here in the middle, we will talk about a process that is antigen presentation. So the rest of this presentation will be divided in innate, antigen presentation, and adaptive immunity.
Okay.
I think many of you have heard that the innate immunity is unspecific, and while the adaptive immunity is a specific immunity. Is that right? Is that correct or no? Edison. Edison is also a ghost assistant.
He's a ghost professor.
Yeah. To Helen? No. Well, when you read that the innate immunity is unspecific, that is incorrect. It's not inspecific. It's non-dependent of antigen, while the adaptive immunity is antigen dependent, is antigen specific. The innate immunity will not react against antigens, but will detect other kind of patterns, for instance, from microbes, from pathogens, but also from signal damage. So you need to have that idea very clear. So, and also we have cellular and humoral in both kind of immunity. I will start with the E. This is the same idea, but with more detail, because when we talk that this is a system, how we define the complexity of a system, both with the elements, but also with the connection among these different compartments and cells. Here you can see barrier function through epithelial cells, mature dendritic cells for monocyte, those are innate, but also we have adaptive cells from lymphocyte, and you can see that they are connected through cytokine, but also for effector molecules. For instance, antimicrobial peptide and antibody. So, cytokine. What is the main role of cytokines? Is the coordination. And we will see that there are cytokines related with pro-inflammatory responses, anti-inflammatory patterns, but also ones that can polarize responses. And for each one, we can see different examples. Pro-inflammatory cytokines, the most common ones, for instance, interleukin-1 beta, TNF-alpha, they are more related with activation of the response. But as I said before, the response can be very aggressive. So you need the other part of the response. So those are the anti-inflammatory ones. For instance, interleukin 10, Tigre factor beta. And I want to mention interferon gamma because this is pro-inflammatory one. And while the others can be produced by several cell types, interferon gamma in fish, well, in other organisms, is more restricted. Why? Because in the presence of these cytokines, macrophage can be really destructive. So the production of these cytokines is very restricted and is mainly to non-cytotoxic cells, because in fish, the natural killers are not fully described, and maybe Th1. So those are the main sources of interferon gamma. These figures summarize the innate immunity. So how we can start to describe this? First, we need a cell. In this case, this is a monocyte. It's really easy to describe because with this shape of the nucleus, we know that this is a monocyte. And how the cells can detect a signal. What is the main component here? Victor, I know you are there. another goes. Yeah, first, we need receptors. And in the case of the innate immunity, these are the pattern recognition receptor, the PRR. There are different kind of T receptor, and they can detect the patterns, the different pattern. For instance, there are specific one detecting a pattern from viruses, other from bacteria, from fungi, but also they can detect dams, and those are damage signals that can be produced by the cell. So the activation of these kind of receptors at the end will induce, will trigger the response through adaptive molecules. And at the end, what is the main focus of everything? Modulating the transcription of the gene. So at the end, we will see the activation of different processes. For instance, inflammation. Inflammation is one of the most relevant process of the innate immunity. So again, we will have this balance between pro and anti-inflammatory molecules, but also the production of the effector ones. For instance, antimicrobial peptide or reactivity species, the oxygen or nitrogen. Next one. The same, but with details. For many people, this kind of figure can be a problem, but you need to start to see what are the main regions. First, membrane, then we have receptors, but you need to be careful because this figure is from higher vertebrates. In fish, the LR4 is not so linked with LPS. And there is many reasons for that. So people describe that fish are more tolerant to LPS, and we need to think why. Fish lives in the water, and the water can be full of bacteria, or there are many, a lot of biomass. So if imagine that they react too much against LPS, the inflammation will be constantly. So there is some people proposing that fish are more tolerant against LPS. I don't know, if you work in the lab and you start to screening different components to trigger the immune response, maybe you need to use high concentration of LPS compared with other, I don't know, flagellin or peptidoglycan. But the idea of this, that fish has many receptors, even more than higher vertebrae. And still there is a lot of research trying to determine which specific ligands are detecting in fish. But not only extracellular signal can be detected, also there is signal that can be intracellular ones, and they are detecting through the PRR linked with the innate immune mechanism. So don't think that only extracellular one are linked with the innate and the other with the adaptive. That is not right. Can be both, depending which is the receptor, is inside or not an endosome. You need a extracellular region in the membrane, so it depends on that. But at the end, we can trigger a differential expression of the genes, and at the end, we will show different immune mechanisms. Same idea, but in this case, with beta-glucan, mannans, and different sugars. In higher vertebrates, one of the main receptors is tectin-1, but in aquatic organisms, there is some people say that it's described, but maybe it's not linked with the detection of the beta-glucan. Can be also not like receptors. So again, we need to be careful because many things are described in higher vertebrates, but when we try to apply to fish, there is some difference. Even though the function, we can determine similar functions, but not with the same components. Some examples of different MAMs or PAM, for instance, in the wall of gram-negative bacteria, we can find the lipopolysaccharide, the LPS. So that is a small region of peptidoglycan. That is the difference with the gram-positive. But not only these huge macromolecules can induce the innate immunity, also some proteins like flagellin. Flagellin is a very special case because the other are bigger macromolecules, but flagellin is just a protein. And it's a protein that can be detected as a PAMS but also as an antigen. So can trigger both, depending on which kind of receptor or which kind of mechanism or pathway are involved, and also time. We saw that in the previous slide that time is critical. Fast and early response, more linked with the innate immunity, while a longer period of time, maybe a higher coordination with needed for the adaptive one. As I say, flyalin is a really good case, a good example, because it's detected by the TRR5, toll-like receptor file, and in fish we have membrane, but also soluble form. So both can modulate the response of the host. CpG, these are linked with nucleic acid from bacteria, can trigger the innate immunity. And other nucleic acid, for instance, from virus, I put also the example of poly-IC. This is synthetic one, but can mimic the molecule from virus. So if you need to work with this, poly-IC is a really good example. And here we can see which kind of virus, if they are double strain, single strain. So depending on this kind of molecule is what receptor will be activated and which kind of response we will get. That is why when we talk about innate immunity, even though some people say, it's unspecific, no, it's not unspecific. It's not detecting specific antigens. Yeah, it's not the same. Because how we determine the specificity of biological control? For sure, one is the antibody complex, more linked with the adaptive immunity, but also we can define a specificity through a ligand and the receptor and the enzyme with the substrate. So these three can determine specificity. So be careful when you say that the innate immunity is unspecific. From fungi, at least for us, they are really good because we are working a lot with functional feed and we are using a specific bioactive from fungi, mainly beta-glucans, but also manoproteins that can be detected for C-type lectins and trigger the immune response. As an overall view, here you can see again the same idea that you need receptors. This receptor will detect a signal. The signal can be extra or intracellular one that can trigger adaptive proteins and different signaling pathways, the activation or regulation of the different transcriptional factor, and at the end we will see a modulation in the gene expression. So this is gene expression depending of the pathogen, because if you work with pathogen, they can always avoid the immune response, and that is other part of the picture. But at the end, what is our main goal? To modulate the phenotype, because at the end, that is the part that is responding against the problem. Question until here. So right now, we can summarize about the global view of the immunity with the innate and adaptive mechanism. We know that time is important, and linked with the innate immunity, we need receptors, we need adaptive proteins, global transcriptional factor, different PAMs or MAMs can trigger different receptors. But which kind of mechanism can we modulate with this signaling? For instance, phagocytosis. Phagocytosis is a really important one. And what is the main concept here? Detect and destroy. How you detect? Through the receptors. After that, you will see a change in the cytoskeleton of the cell. So at the end, the cells can trap or can get the macromolecule, and then we will see how this structure can form a phagosome. For the one that are working with salmon, this will be really important because in Chile we have the problem with Pizzerichetta salmonis, And here you can see different mechanisms to avoid the immune response, because in this one, after the phagosome, you need to be the fusion with the lysosome. The lysosome will have all the enzymes and the respiratory burst to destroy the bacteria, and but in the case of Pichirictis salmonis, they can survive this, so they can avoid the respiratory burst of the macrophage. At the end, they will survive there. So how you kill something that is inside a macrophage? So that is why it's a really good pathogen. So in this case, after, if the process goes in a good way, we will see the digestion, the destruction of the problem through enzymatic response, the production of reactive species. But then we have something here that is really relevant, is the antigen presentation. So at the end, this will be the bridge with other kind of immunity. And that is why also the adaptive immunity did need more time. So it's from the innate, the activation of the immune response, that then we can coordinate the adaptive one. After the digestion, also we will see the production of cytokines. So at the end, we will see our inflammatory response, mainly related with M1 macrophage, but also we have the M2 that is our wound healing. So at the end, we can see the balance. Again, this idea of maintain homeostasis is not just activation, also related with control. The idea that I was telling before, so we have different issues, injuries or infection. We have different mediators. And at the end, we will see healing and resolution. These are more linked with the M2 profile. Chemiotaxis is when you need to mobilize different cells. For instance, interleukin-8 is a really important chemokine. Linked with the production of effector molecules in the innate immunity, we have the respiratory burst. But we need to be careful with this one, because through the production of reactive species from nitrogen, oxygen, yeah, we can destroy a pathogen, but also we can destroy to the host, mainly to the disruption of the DNA of the host. So again, this molecule has really short-term life and can be modulated and can be controlled by all the antioxidant system or antioxidant response from the host. So for people that is working with antioxidant component, you need to be careful, because one thing is the antioxidant ability of the specific component, but also the fish has their own antioxidant capacity. So you can modulate both. This is the antioxidant response, here mediated by antioxidant enzymes, superoxy dismutase, catalase, and GPX, but also peroxyredoxime are very important in fish. And why? Because depending of what is the structure of the protein will be the function. For instance, peroxyredoxime has antioxidant capacity when they are in a dimer, but in the camer, they have chaperone function, or even only in single form and monomer, they can be secreted, a natural killer enhancement factor. And at the end, we will see the activation and the crosstalk between macrophage and cytotoxic cells. So again, I think the key concept today is balance. So antioxidant control, but also I can activate other cells. So always remember this idea of homeostasis. Here we have a comparison between mammals and fish. So this idea that we have M1 macrophages, they can be classical or innate, and M1 are more related with activation, more aggressive response, while the M2 are more linked with the balance, the wound healing process. And that is, well, still there is some gap of knowledge related with this one. Are the same population? Is one macrophage that can be both depending on epigenetic features? This is still on discussion. But again, we can see as a balance between activation and control. And this balance will be linked with the cytokines because we already know that cytokines coordinate and then also can be related with effector molecules. And for instance, for M2 is really important arginase. That enzyme can be linked through the metabolism of arginate, preventing the production of reactive species of nitrogen. I already told you that if you read humoral, is the fancy way to say molecular. So humoral response, in the case of the innate immunity, that will be linked with the complement. And the complement has different ways to be activated. Can be classical pathway, lectin pathway, or alternative. But again, this depends is how was the activation. The classical pathway is through antibody, antigen-antibody complex. So here you can see the connection between adaptive immunity with the innate. Because which cells will be detecting this will be granulocyte, for instance, and they are innate. For lectins, they are the detection of sugar, for instance, mannose, and for alternative pathway, that will be pathogens and other kind of molecules. But at the end, all of them induce this part of the complement, C3 convert. And then we have the cascade to, at the end, produce the MAC complex. And this will be linked with cell lysis and activation. Questions? No question until here? We have already 35 minutes. A lot of it. I will start to finish with this one first to talk about other type of human immunity. This the antimicrobial peptide, for instance, cathelicidin, hepcidin, that can induce the disruption of the pathogen, and they are very important, and they can be produced really fast. Something that is really new in aquatic organisms is training immunity. Why? Because this suggests innate memory. If you read about immunological memory, I think mainly the information will be linked to adaptive response. So it's memory against the same antigen. It's a better secondary response. But in the case of training immunity, this will be linked with the innate mechanism. And it's really good because the second challenge is not the same that the first one. So you can trigger training immunity, I don't know, with LPS, with beta-glucans, but then as a secondary challenge, you put another problem, another pathogen. So with that, you can have a wider or a bigger response against different problems. So that increase the possibility to the system to react and improve the survival of the host. The difference between training immunity and a regular immunostimulation is this part of the response. Is the resting period. If you remove the signal on the stimulus, you will see the response going back to a baseline. For immunostimulant, the response will be increasing until re-stimulation. But for training immunity, you will see that you remove the problem and the response goes down. Still, this is under evaluation, there is a lot of people trying to describe this because how much we can wait without lose the response. So this is a really hot topic right now. Overall view of the innate immune response. Again, cellular responses linked with barrier functions, epithelial cells. We have phagocytes. We have other cells. Also, we have molecules. So again, innate will be able to detect pathogens or microbes or danger patterns signals, and then we will trigger the response. The response at the end will be cellular or human. So after this, we have this bridge. The general definition that this is the bridge between innate and adaptive immunity. In fish, still, that is something that need more discussion, mainly related with which specific cells are professional antigen-presenting cells. Because all cells with nucleus, they can present antigens through MHC1, but also we have the professional ones, and those are for MHC2. And we will discuss some examples. This is the overall view of the antigen presenting cell from mammals, and we will see dendritic cells. These are in fish, are not fully reported. So people are proposing dendritic light cells. Macrophage also can present antigens, and the same from B cells. But that is different between them because at least in higher vertebrates, only this one can induce the activation of naive cells. And that is the difference, because that is antigen presentation to naive cells, but also to effector subtype were already activated. So that is something that in fish need more information. Which are the antigen presenting cells? Here we have the professional ones, B cells, and DC, the dendritic cells, and the macrophage. Those are more innate, and these are adaptive. The problem of dendritic cells that they can present antigens in an immature or a mature form. That will impact the type of a presentation, and this is really relevant. For many people working with vaccine, they never care about this, and this can make a lot of changes. For instance, if the dendritic cells or the professional presenting cell is immature, they can induce tolerogenic responses. So at the end, you are putting an antigen, and the animal will be inducing a tolerogenic response, a controlling response, inducing immunosuppression. While a mature cell with more co-stimulatory signal, they will induce a more strong presentation, and maybe that will polarize the response to other effector cells. How is the presentation? Mainly through two mechanisms, MHC1 and MHC2. MHC1 is when the antigen is intracellular one, and MHC2 is extracellular one. But of course, in biology, we always have all the possibilities, and also there is cross-presentation. So the system always can be worse. Question about this? No? I will put more detail in the next slide. These are the different to determine which kind of MIC class, 1 and 2. You can see difference in the structure of the protein, which kind of subunit has. And IC way is how many transmembrane segments they have. MIC1 is only 1, in the others 2. But this is part of the cell that is presenting. What about the cell that needs to respond? And that will be a T cell. And the T cell is T because they have TCR, T cell receptor. So this TCR will be able to detect only one kind of antigen. And that antigen needs to be in a complex with the MHC. Otherwise, this T cell needs to be induced to apoptosis. They cannot detect the MHC complex alone without the antigen. Otherwise, we will induce we have all the autoimmune diseases. This is the relation between both. Here we have the professional antigen presenting cell, and in this case, the T cell. So we have different signals. Everything starts with a receptor detecting something, then we will trigger all the response that we saw in the previous slide, and then we have First, a connection by touch is a juxtacrine communication. So it's through the MIC peptide complex that can be detected by TCR. And TCR will be stabilized by CD3. Then we have co-stimulatory signal, in this case CD86. So this is signal 1, signal 2, and the environmental one related with cytokines will be signal 3. So this is a kind of antigen presentation that will induce a really strong response. And we know that this can happen without these ones. For instance, with a lower level of co-stimulatory signal, at the end, this can induce different ways in the activation of the T cells. Because the T cells, after this, they have different roles, can be Th1, Th2, and Treg. Example of co-stimulatory signals, if you want to read a little bit more. The same, depending on which environmental signal will induce the presentation. And here you have the comparison between mammals and the potential field component. So in this case, is the example for Th1, cell-mediated immunity, so specific against a specific antigen. Innate cells, adaptive cells, adaptive subtype, and at the end, adaptive response. The same idea, but with other subtype molecules. Th2 is this cell then will be involved in the coordination of B cells, and at the end, B cell will produce antibodies, and we will see different populations. Th17, more related with pro-inflammatory patterns, and at the end, regulation. Again, the idea that is not only important activated, but also control. Here you can see specific samples in fish. This is the Bible is one of the best report in fish to describe dendritic-like cells. And here you can see how the progression of these cells from monocyte to dendritic-like. This is the cell that I already described, and these are the ones that are acting as a professional antigen-presented cell in salmon. These are the co-stimulatory signals. This is a marker for dendritic cell maturity, and this is in fish. This is a global transcriptional factor that induces the progression of these professional cells, and MIC2, so it's a professional one. More info, this is phenotypic characterization. You can see here the cells with the proteins in the membrane, and then some result at flow cytometry. Data from the field, because this is not just described in the lab, also you can detect this, this kind of pattern at field work. This is an experiment in Chile, so against Pseudocytes salmonis. The previous results were related with the antigen presentation, but this is T cell polarization, so the other part of the process. And in this case, even though we put interferon gamma, and in the previous slide, so I told you that interferon gamma induced an aggressive response in macrophages. In this case, we polarize the response to Treg. So this idea of the balance, always need the balance. Who is controlling this? In this case, FOXP3. This is a global transcriptional factor linked with the regulatory cells. And at the end, we can summarize in this. So we talk about innate and we talk about antigen presentation. The last part of the process is the adaptive immunity. Again, from a mammalian point of view, we have cell-mediated immunity, for instance, linked with the Th1, that is a crosstalk between these T cells with macrophage, but also we have the cytotoxic T cells. In the other hand, we have the humoral immunity, and in adaptive responses is linked with antibodies, antibody production. Different classes of lymphocytes, B cells, the helper, the T cell, cytotoxic one, and the regulatory. If you see this, mainly is related, you can compare with training immunity, but in this case, it's adaptive one, and this is the process of the activation. So we will see that the antigen presenting process, clonal expansion, differentiation, and at the end, after the activation, we need to see apoptosis. But in adaptive one, we know that will be some survivor memory cell. In fish, compared with higher vertebrate, this is under evaluation and also how much time they can be available. If you saw this figure again, we saw an increase in a fair response and then a better secondary response. But in this case, different to the training immunity, this will be antigen dependent. So will be against the same antigen. That is the difference with the training immunity. A little bit of the clonal expansion. So you need to know that each lymphocyte is able to detect a specific antigen, and depending of the antigen presentation, that will be the selection. Then we'll have a clonal expansion and a selection, and at the end, in this case, is linked with B cells. We saw plasmatic cells producing antibodies. To summarize, linked with the naive B cells, a couple of them can be regulatory. Also, we can see the ones that are producing antibodies. A little to talk about antibody structure, we can see constant region, variable region. In the variable region, we can find the antigen binding site. So these are the part that can detect the antigen. While in this part, then we can see that some cells can have receptor against this fraction. So then we can have innate cells, but with antibodies in the member. And with that, again, the coordination of both mechanisms, innate and active. At the end, we are inducing an integration of response that will be more robust. And with that, their time of response will be shorter. So we can modulate the survival of the fish. Different types of antibody feature, but I want to talk a little bit about this because we have difference with the mammals. In fish, we have other type of immunoglobulin. For instance, IgT. IgT has some functional similarities with IgA in higher vertebrates. This is more linked with mucosal. But still, one of the most relevant one is IgM. Here is the proportion of the different antibodies, but with a higher vertebrate point of view. And I think that is everything. A lot of information and not too many questions.
Thank you, Dr. Byron. Gracias por la presentación. Vamos a ver si es que de nuestro público